So, hey, we're group number 13, and my name is Milda, and my group mate is Karen. And we're both PhD students from Linköping University, though Karen is also industrial PhD student from Sectra. And our research is focused on deep learning applications for digital pathology. So this is a research working with medical imaging. That's why genomics data is a very new field for us, and it was a very exciting challenge to look how we could do analysis using this type of data. Our project is called Genomic Analysis with Glow and Spark, and we wanted to see if we could see or determine correlations between human genome and different ethnicity groups. This work was inspired by a paper by Huang et al., which is called Genetic Differences Among Ethnic Groups, and it was published in 2015. And also some code in the notebook was borrowed from a genomics library called Glow, which is implemented in Spark by Spark. Yes, so background of the problem. As we know, each individual or each person is kind of encoded using DNA. And DNA is a structure of nucleotides, and it's structured into double helix. And there are 22 chromosomes that each of us has. And chromosomes, or these DNA, it's made out of 4 nucleotides denoted by A, T, C, and G. And most of the DNA is actually identical between the human beings, and only a small proportion is varying. And a common SNPs are representing these variations. It's variations in 1 nucleotide, and they encode all the differences that, or most of the differences that we observe between the people. So different skin color, different eye color, different height, and so on. This analysis was actually very expensive for a long time, and because the sequencing cost of human genome was very expensive. But as we can see from this plot, the cost started decreasing sharply from 2007, and this enabled researchers to collect quite big data sets of human genomes and sequence, and those data sets are publicly available, and one of them we used in our research. And now Karen will continue forward for the introduction.
Yes. So the data that we have used is from a project called 1000 Genomes Project. And that was conducted between 2008 and 2015, and where a lot of genomic informations were collected from people all over the world. And so the data that we are using is actually only originating from 1 chromosome instead of all 22. This is done for simplicity, but even so, the data that we have is approximately 6.5 million SNPs from about 2,500 different individuals. So what we did in this project, let me just break it down a little bit for an overview for you. So we start by reading the data. Genomics data is stored in a special file format. We'll see that in a bit. And then we needed to do some filtering because not all of these SNPs are high quality ones. Some problems can occur while you're doing the sequencing, for example. And we're also doing some analysis with PCA and so on. And then our main aim of this project was to see if we can find a correlation between the SNPs and the ethnicity. So we're doing a linear regression analysis and also using another method, right, Milda?
Yeah, the random forest.
Random forest, yeah. So let me just go through the code here and see what we did. So first of all, we have our libraries that we include. And let me just say also that we're not going to run our notebook live here because there's a lot of cells that take a lot of time. So I'll just go through it. And as you see, we've written it in Python as well. And the most important libraries that we're using is this Glow, which is included if in this Databricks for Genomics package that we can include in our cluster. So we are using some helper functions just for plotting, basically. And here we just specify some paths. And the data that we got, we downloaded from this website. And now we want to read it. And as I said, we're using this GLOW package here. So once we read the data here, it's in this file format VCF, and then we can, using Spark here, we can read it, and then we can get it in this more nicely formatted way. Oh, sorry, it's jumping around a bit. So what we see here that for each row, we have one of these so-called SNPs, and we can see where in the chromosome the position of this specific SNP and what the nucleotide is, both for the reference and for the other people in the data set. So what is actually the most important column for us is this genotypes. And here we're only displaying the genotype for the first person sample ID HG00097. And so this is the most important part, the 0 and 1, or 0, 0 or 1, 1, which gives the genomic information, basically. So this is what we want to extract. And this is available then for each of the SNPs. So now we have approximately 6.5 million rows in this data frame. So we want to filter that, basically. What is very common when you're handling with the genomic data is to do some just basic data manipulation. For example, we want to do this split multi-allelic function that we're using from GLOW, which is basically a way to just reformulate the data so it's structured, like so that we can make sure that on each row we have a unique SNP, basically. And then we want to calculate some statistics, like how much, like the occurrence of the genotypes, but also this Hardy-Weinberg equilibrium p-value, which is also a statistical metric that is commonly used for genomics data, which basically tells you the probability of a given SNP variation, which is called allele, how probable is that this variation actually occurs, or that it's a mistake from the reading. So by calculating this statistics, we can then filter out low quality alleles that are less likely to be true ones. And we also filter those that are like based on the frequency of them as well.
Okay, so now we have prepared data and filtered most SNPs out. And the next thing we want to do is to look if we can see any correlations between ethnicities using PCA analysis. The downside of PCA analysis is that we cannot directly connect SNP to which SNPs actually explain the ethnicity, but it does give us indication if there is any reason to believe that there should be some structure in the data. So we use the data that Karen explained how we got it, and we apply the PCA analysis, and we can go a bit down. Thank you. Yeah, so it's basically just standard functions to apply PCA, and in the end we get the plot, which I think is the most interesting part. And we look just at the first and second principal components in the plot. The colors are the different ethnical groups. So we can see, and each point is kind of representing the point in data set. But of course, as I mentioned, that PCA is not allowing us to connect directly which SNPs are contributing to predicting all these ethnicities. But the interesting thing that we see is that even though the structure is not perfect, but we do see clouds that the different ethnicities are grouped in different spatial locations. And this is consistent with the previous research that also had these findings. And this is what inspired people to go one step further and do correlation analysis, trying to figure out which exact SNPs actually contribute to the ethnicity differences. And this comes now, it's the part about our main goal of this project. So we wanted to replicate parts of the results from the paper that we mentioned in the start, which is called Genetic Differences Among the Ethics Groups by Huang et al. As we also mentioned in the beginning, we started with 6.5 million genetic variations from 2,500 individuals, but then we did a lot of filtering and now we have 400,000 variations, which seems to be quite few, but in reality, the research, and in this paper that we're citing, it's also mentioned that much fewer SNPs are actually related to the ethnicity. And that gives a rise to our hypothesis that we should be able to filter even more out SNPs and still get good performance on the two models that we apply, so logistic regression and the random forest. And that is exactly what we're trying to do here. So we take our data and we can go a bit down. So first of all, we just plot the data. We got the data about, for each sample, what ethnicity that person belongs to. And we just plot a bar plot of his data. And as we can see, the data is not really balanced. We have people with African origin, they're dominating the data set, but we decided not to balance the data and just see what results would be. And if our results would be very poor, then we would be considering if we should balance data or do some other solutions. So now we can scroll a bit more down. And this was, we were just testing one hot encoding, but I think in the end we realized that we don't really need it, but because, as Karen mentioned, all code takes to run very long, and because we included it in one of the data frames later, we kind of, for completeness, we left it here. But it's, yeah, in some sense it's redundant.
Yeah, but this is just a nice way of just highlighting how each sample, then the labels that we have for each person, and then for each person we again have a lot of SNPs. So here's a sample ID and then just a superpopulation that we're using. So this is a European person.
Yeah, and of course we need to index it to numeric values for our analysis. So that part is actually relevant population index column. That European was indexed as index 2 and so on. Yeah, so in the paper that we're basing our analysis on, they say that 85% of SNPs are basically identical in human populations. And we need to select somehow those only important ones for ethnicity. And in the paper, they actually use a different technique than us. They use Kramer's V statistic, but unfortunately, we could not find it implemented in PySpark or in any of the libraries that we could use. And we decided to use a chi-square selector because it's the most similar feature selector that we could find. It's based on the same distribution. And in our experiments, we decided to pick different subsets of SNPs based on this selector. So we would pick the most important. So first we took all of them, all after the filtering. So it's around 400,000 SNPs from chromosome 1. And then we were filtering using chi-square selector to 200,000, 20,000, 2,000, 1,000, 150 SNPs. And our hypothesis was that for some time with reduced amount of SNPs, the performance should still be good, as important SNPs are not all of the SNPs that we have. But when we reduce too much, the performance should obviously start falling as we start omitting some SNPs that are actually very important to predict the ethnicity. So the code below is, we had to load the data, we had to match the ethnicity to the SNPs that we have, and then we have to format the data, transform the data in a scalable way that would be possible to run analysis. And actually these steps are, especially analysis steps, are inspired by the labs that we did for this course before in the lab 1. And yeah, in the end here we get, so we needed to use functions expand to expand SNPs and expand states. And if we go down, we can show the final version, what we get. Yeah, so this is our data. So we have a sparse feature vector and a population index. So population index is for this what kind of population it was. And population one hot encoded was actually something we didn't need, but we were not sure about that. And because like each computation took so much time, in the end, we did not fix it. And now on this data frame, we can fit a chi-square selector. And as you see, maybe I did not explain very clearly in the data frame above. So the first column is actually our 400,160 blah, 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 SNPs. So it's how many data rows we actually had, but now we congregated them into one sparse vector per each observation. Okay. And so chi-square selector, we just imported it from PySpark machine learning library. We commented it out because it did take to run very long. So for each different subset of like how many SNPs we want to keep, we did a separate cell and we ran it. And then we saved the results to disk and we loaded it. And in the end, we had, so we did all of those that we listed above. So from 200,000 to, I think it was 50 in the end. So this is like for 1,100, and then 50 was the smallest subset that we used.
Yeah, and this is how they ended up.
Yeah.
Like this is the column.
Yeah, we can see that instead of those 400,000 around features, we only have 2,000 in this example. And these are the most important selected based on chi-square selector. And now this is the part where we apply the logistic regression and random forest. As I mentioned before, this is inspired a lot by the labs that we've done before. And we use metric accuracy that we compare our results. And so we fit random forest and logistic regression. We fit on each data set. So as I said, we had full data set, like a bit over 400,000 SNPs. Then we had 200,000 SNPs, 20,000, and as you can see here, all of them. And we just did a loop here and we just, fit the model and then calculated the accuracy of the prediction. And finally, we plot the results, what we got. As you can see, logistic regression is the blue dots and its accuracy, and the red dots are random forest. So logistic regression outperformed random forest for all of the data sets, like subsets of the data sets that you tried. And it actually achieved very high performance. It's above 90%, which really confirms the previous research and our hypothesis that SNPs do contain information about ethnicity. And also what we can see that even when we reduced the number of SNPs quite a lot from 400,000 to 20,000, the performance was reduced very little, very insignificantly, like the first even 3. So that also confirms the hypothesis that not all SNPs are important for ethnicity and you can predict it from much fewer points. Of course, when you look at random forest, not the same tendency was observed, but random forest in general performed worse. So we can make a conclusion that maybe it was not capable of really capturing the correlations between SNPs and ethnicity. And now Karin can sum up and give some overview.
Yeah, so just some final notes about our project. As you probably all understand, due to time limitations, we reduced the scope of our project just to include data from chromosome. We limited the way we filtered it to kind of like just do the bare bones. Also, in terms of analysis, we only tried 2 different methods, but again, we did try many different numbers of included SNPs. But continuing to work on this, you can definitely tune hyper parameters or add confidence intervals, do training with more models. And as in this paper, instead of just using chi-squared feature selector, we can do something else that perhaps would be able to better capture the relevant SNPs. But overall, this has been a very interesting project. As we said in the beginning, we didn't really know anything about genomics data, so we took some time to read up on both the problems that can be solved using these types of analysis, and also how the data is formatted. We had to struggle quite a bit with the fact that each row in the data set is a SNP, and for each row we have 2,500 different values for each person, which is not really how we want to have the data formatted for this type of, for example, the logistic regression, where we want each label to just have one input instead of one SNP having 2,500 inputs. So we had to format the data quite a lot, which was challenging, but rewarding in the end. So I hope that you found this project interesting, and well, thank you very much.
Thank you.